Well, I'm going to open up with a question that's really been inspired by the conversations that I've heard today. Ruslan and I had a chance to talk about this a little bit at lunch today. I've heard, in the last session, we heard people talking about the issues of doing correlative work and the potential need to do correlative work in the era of big data and big data analytics. And on the opposite end of the spectrum is the ability to and the need to use model systems to look at the mechanism for what might be driving the composition, diversity, and function of the gut microbiome and the implications for human health. And so I see this spectrum of approaches being reflected in our panelists, so it would be great to get everyone's brief thoughts on how do we synergize these different approaches that appear to be on opposite ends of the spectrum.
Yeah, I think this is a very difficult question. You know,
I should have started off with something easier.
Yeah, no, you know, I think that you're right. This has to be a synergism between the you know, human-driven studies and mouse models for mechanism. I think we may be lucky in some respect in that we can reconstitute a human microbiota in a germ-free mouse, and it faithfully reconstitutes. There are some differences, but we see, you know, the major groups are the major groups, and we see the major metabolites are the major metabolites when they go into a mouse from a human. And so this gives us a great opportunity to study a human microbiota in a microbial context. Now, when you start extending this to host microbiota interaction, it becomes a lot more complicated because of the coevolution of microbes with their hosts. And particularly in respect to how the immune system or how aspects of metabolism or even the central nervous system, how that responds to the microbiota. You know, we'll have to take this on a case-by-case basis to see in what situations the mice actually serve as good models for what we want to study. But we know, for instance, it came up this morning with the antibiotic panel, the issue of microbiota deterioration over time, the suggestion of multi-generational experiments in mice. And that's something that's just really hard to do, this idea that over sequential generations we're losing microbes, and the signal is incredibly small from one generation to the next, but over millennia, really significant. Okay. And so in mice, you can go into this and test this, but I'd be curious to hear Ruslan's thoughts about connecting this back to host biology.
Well, so I think it's an issue that I think is a major challenge for the entire field of microbiome study, in that it's a system with unknown degree of complexity. The enormous progress that's been made in cataloging the complexity through new modern technologies has been eye-opening. It told us about this, not just huge complexity, but some aspects of organization of this complexity into microbial communities and their connections through various well-known ecological mechanisms. But the problem is we don't know what degree of that complexity is relevant to the host. Depending on what you ate for lunch, microbiota would be different. Depending on where you travel, it would be different, but you know, you still overall would be the same person with probably at worst some mild stomach issues. But generally, it's not going to have a huge impact as far as we can tell. So even though there would be huge shifts in the composition of microbiota. So it's enormous complexity. We don't know how much of that complexity is relevant, and we don't know how to handle not just the big data that is generated by these types of approaches, but also how to deal with the problem of causality versus just correlation. And I think it's what it calls for is not just improved computational methods, not just improved statistical handling of the data, which is sufficient in many other fields that deal with big data. What it calls for is developing a new type of concepts and knowledge that would allow to make determination if something has biological relevance and what sort of what kind of biological relevance, whether it's organism specific or disease specific and so on. And this is something that is, I think, is way behind, not surprisingly, because generating big data now is becoming much more feasible, much more doable. But developing these ways of understanding how that works is behind. And I think something we can learn not just from ecology, but also it's similar problems were faced, of course, by sociology over the decades. And sociology had many dark moments when big conclusions and inferences were made based on not just on correlations, but on things that are on the surface, something that's easy to detect, something that's easy to make correlations with, and I think the field of microbiome should try to avoid that.
Amanda, what are your thoughts?
I apologize for interrupting. I can't really add much to what Ruslan was saying, that we do need these studies of various populations and various humans to sort of document and catalog the diversity that's out there. But until we understand what the exact function of it, what do these mean? Is it meaningful to the host that certain species are there or not there? That this information is curious and interesting, but not directly useful for the things we want it to be useful for, studies of human, therapeutic use. So the kind of working with mouse models, working with controlled environments, and just doing enough of these studies so that you can try to control for some of the factors that might be at play, whether it's host genetics or certain lifestyle types, whether it be subsistence farming or pastoralism or even hunter-gathering. Looking at more groups who practice each of these, you can try to tease apart how much of the pattern we see in the gut microbiota is predicated on those differences.
Thanks, Amanda. So it looks like I have time for one more question before we open it up to the audience. And I wanted to share a question that's really derived from conversations that I've had with Brendan Bohannon at the University of Oregon, who was a moderator for a panel earlier today. He's a collaborator of mine on the built environment work that I do, and Karen Gilliman at the University of Oregon, who uses model organisms, zebrafish and stickleback, in collaboration with Bill Kresko, to understand the mechanisms that drive microbial community assembly and host health. Something we've been talking about is a dimension of diet that often gets lost in the conversation, and that's of food preparation. And Amanda, you might have some thoughts about this, but the practice of cultivating, or basically farming foods indoors, we've lost that with the advent of the refrigerator. So we vary, as an example, fermentation used to be the primary way that we would store our food. And as a consequence of this, not only are we ingesting food that has a much different and less diverse microbiota, but we're also inoculating our homes and our kitchens we're missing that inoculation process because we're not farming microbes indoors anymore. So I was wondering if you could all give some brief thoughts on how you think, how important you think food preparation is to the gut microbiome.
Sure, we can let Amanda go first.
Amanda?
Thanks. Yeah, absolutely. This is such a huge question, and I would argue that there's two ends of this spectrum. You can go back in human history and ask the question, how important was cooking in terms of reducing microbial load on the food that we consumed? You can bring it up to recent times and ask how important is the sterilization of most of our food, the highly processed nature, not only removing this dietary fiber, but also intentionally removing a lot of potentially internal bacteria might be for changing our microbial communities. it also begs the question, how important is it to eat food that is from the local environment in which you live? I mean, granted, most of us are spending 90% of our time indoors, but we don't have the connection to the land upon which we live anymore. So this is an area that's never been explored, to my knowledge, and could be very productive. And maybe bringing back some of these fermented foods, or considering consuming foods that are less processed or locally grown, all of these things may have a positive influence on our gut microbiota.
Justin, do you have thoughts?
Yeah, I think there's, if I could break the question into two different parts, I think one is the farming aspect, and then the other one is just foodborne microbes in general that might be present on roots that are dug up and consumed. And certainly our food supply has become virtually sterile, and we used to consume a lot of microbes, I think. And it's remarkable that you know, the field of probiotics is gradually shifting into legitimate science, and there's some really nice findings that are coming out of that area, and perhaps it's not too surprising because these organisms that are used for probiotics are found in fermented foods. We're essentially supplementing those back into our microbiota and essentially complementing this microbiota that has, this portion of our microbiota that has been lost. So I think it's very likely that you know, fermented foods and the microbes that occur on them are an important part of human health, that we've you know, co-evolved with these to some extent. And then the foodborne microbes, you know, this has been a great benefit because we've lost all these pathogens, but in the process maybe lost important, you know, soil-borne microbes that play important functions. There's a really beautiful example of genes coming in from the outside from a marine bacterium laterally transferring into the gut microbiota of Japanese individuals, a study published in Science, enabling the microbes in the gut to consume seaweed. And so the microbes from our environment actually can be an important repository of genes that are useful in helping digest food as well.
Ruslan?
Well, I don't have a lot to add to this, but I think we, like everything else, we need to consider this question in the context of all the pros and cons and specific conditions where particular types of diets exist and particular types of cooking and so on. There are many benefits of cooking, of course, of cooking the food that we consume. There are some, it can be argued like as a general question to what extent something that's natural is good for you and something that's not natural is bad for you. It's a very important question. There are many, you know, we intuitively feel like anything that's natural should be good, and it's true for many things, but it's also natural to, you know, to have Hellman's infection and die when you're 30. That was very natural 100,000 years ago. It's no longer natural, and I think we all can live with that. And so there are lots of things that we can extrapolate this way, that based on the fact that there are many things that are very unnatural and very bad for us, and to extrapolate them to everything that is natural might be good for us, and it's not always the case. And with cooking, I think it's a balance of a variety of factors. You cook and you use clean water and you free basically of parasites. That is a great news, but it also brings some additional, potentially brings in other problems, as Justin mentioned earlier, potentially predisposing us to certain types of inflammatory diseases. And I think it's the type of a question that, again, requires very specific controlled studies, but the answer is likely to be that what's good for Australian Aborigines may not be good for Eskimo, and that may not be good for a New Yorker. So it's something that needs to be considered in the ecological context.
Thank you.